to our third speaker of this afternoon. It's my pleasure to introduce Dr. Laura Crucianelli. She's a Marie Curie Research Fellow at the Department of Neuroscience at the Karolinska Institute in Stockholm. She completed her PhD at the University of Hertfordshire in the UK and her first postdoc at the Research Department of Clinical, Educational and Health Psychology at the University College in London. She worked with Paul Jenkinson and Katrina Fotopoulou. Now she's currently working under the supervision of Eric Herson as part of the Brain, Body and Self Lab. Today she will talk about some of her previous and earliest work, giving a talk title Comfortable in Our Own Skin, the Perception of Affective Touch in Anorexia Nervosa. Thank you, Laura.
Hello, hi. Can you hear me well? Are you see my screen? Okay,
totally,
right?
yes.
Yeah. Great. Good afternoon, everyone, and thanks so much, Francesca and Martina, for inviting me. I'm really happy to be virtually in Parma today with all of you and with all these amazing women who I admire a lot. So as you said, oops, hello? Do you still see a screen?
Yes.
Okay, 1 second, because
perfect.
Perfect.
Okay.
So as you mentioned, I'm a Marie Curie Fellow at the Karolinska Institute, but today I'm just going to talk about previous work that I've done at the end of my PhD and my first postdoc with Katrina Fotopoulou. So I actually don't think I need to give any background because you all know a lot about touch by now. So I will just stress the fact that touch is much more than a simple sensorial stimulation on the skin. I'm just trying to cover this topic that I think also Frances McGlone addressed in the questions about the distinction between discriminative and affective touch. By saying what actually happens when something touches our skin. So we know that we have an immediate activation of the myelinated fast-conducting A beta nerves. This is a rapid first touch system which has obvious advantages for our survival because it allows us to identify promptly something on our skin so that we can react in the most appropriate way. In parallel, and only when touch is likely to also have an emotional and affective meaning, there is also the activation of the unmyelinated CT afferent system, which is a second slow touch system, which perhaps has less obvious advantages for our survival, but it has been suggested that it might really provide this neurobiological substrate for the development and function of the social brain. So it has been proposed that we might have a specialized system for the detection of affective touch. How do we know that touch is not all the same? We know a lot through studies that use this technique called macroneurography, which is a very sophisticated technique which allows us to record single unit activity from the nerves in the skin. And thanks to these very important studies, we have been able to identify the preferred range of velocities which optimally activate the CT afferent system. So we know that the system is optimally activating when velocities are between 1 and 10 centimeters per second, meaning that when touch is delivered at lower or faster velocity, there is a reduction in the activation of the system. And very interestingly, when touch is delivered at these velocities, there is also an higher level of pleasantness reported by the participants. So these very important studies published in Nature Neuroscience, as we see in figure C, it shows that there is a linear correlation between the firing of the CT afferent system and the subjective report of pleasantness. So this system has been found mainly on hairy skin. I say mainly because this very important study came out just this year and they were able to record for the first time CT afferents also in the palm, although the ratio is still quite different, so it's 1 to 7. Every CT afferent recorded in the palm, we record 7 on the hairy skin. So still we say that this system is mainly found on hairy skin. And as I said, there is this preferred range of velocities, and there is also a preferred range of temperature. And the preferred temperature is the one neutral, typical of the skin. So if we deliver the touch at cooler or warmer temperature, there is a reduction in the activation of the system. What is very interesting is that the signal from the skin through the spinal cord, it seems to activate the posterior insula rather than the somatosensory cortex. And that's why affective touch seems to share much more characteristics with interoceptive pathway rather than exteroceptive pathway. And that's also why it has been redefined as an interoceptive modality, because it has been suggested that affective touch provides information about the physiological status of the body at any given time. And this redefinition, it was mainly driven by a work, very important work done by Craig. So, very good, we might have this specialized system, but why do we have this system for the detection of affective touch? So we know that it might represent the foundation for affiliative behavior, is a very important mechanism for the formation and maintenance of social bonds. And we also know there is a powerful non-verbal means for the communication of emotions. Caterina has already mentioned this very nice studies coming from our lab showing that participants are very good in detecting emotion and intention via touch based on the velocities that we use to deliver the touch. However, is this the full story? We didn't think so. So we really wanted to investigate specifically the role of affective touch to the way we become aware of our body as our own. So we conducted a series of rubber hand illusion studies. I'm not going to explain what is the rubber hand. I assume you know. So in this study, we delivered the touch during the rubber hand illusion at either slow CT optimal velocities or fast CT non-optimal velocities. And what we observed is that slow CT optimal touch was perceived more pleasant and also enhanced the perceived embodiment of the rubber hand more than fast emotionally neutral touch. Therefore, we concluded that this touch might play a unique contribution to the sense of body ownership and to the way we recognize our body as our own, and by implication to our embodied psychological self. And we were very happy that also 2 other labs replicated the same finding and expand a bit on these findings, and we were also able to replicate this finding a few years later. Now, given the role that this type of touch seems to play to the way we represent our body, we decided to investigate this modality on a clinical population characterized by body image distortions and lack of awareness, such as anorexia nervosa. Anorexia nervosa is an eating disorder characterized by restricting eating and an obsessive fears of gaining weight. The etiology remains unknown, but we know that impairments in social cognition and reward circuits play an important role in the onset and maintenance of the disorder. But there are not many investigations of the relationship between these 2 aspects. So in this first study that we published a few years ago now, we aimed to investigate whether anorexia nervosa was associated with a reduced perceived pleasantness during social tactile interactions. We tested 26 people with anorexia nervosa and 30 healthy controls, and we were delivering touch at slow CT optimal velocities or fast CT non-optimal velocities, and we were asking participants to tell us how pleasant was the touch. There was also a social manipulation with the facial expression, but I'm not going to go into details of that now. What we found is that CT touch elicits significantly lower pleasantness in ratings in people with anorexia nervosa as compared to healthy controls, but we didn't find these differences between groups in CT non-optimal touch. So the deficit seems to be specific for CT touch, and that's why we suggested an involvement of interoceptive perception and a potential link with body image distortions. Again, we were happy the other research group conducted similar work, and in particular, I would like to point out the work of Davidovich of 2018, in which they also is a neuroimaging study, and they replicate our finding, and they also found in anorexia nervosa, as compared to healthy controls, a reduced activity in areas including the left-coded nucleus, and this area is very important, as we know, for the wanting network. So we think that these data really fit very nice. These neuroimaging data fit very nicely with the behavioral one. Now, a question that you might have, that many people have, is whether this distortion in the perception of affective touch in anorexia nervosa is a cause or consequence of the disorder. So it's a bit of the chicken-egg issue. And of course, it's very difficult to address this question, but we try to understand a little bit better what's going on in our next study that we published very recently, and in which we tested the perception of affective touch in 27 women with anorexia nervosa, 24 people who have recovered from anorexia nervosa, and 30 healthy controls. We can have a big debate about what recovery means in anorexia nervosa, and if you want, we can talk a bit more about it later. So in this study, in the first part, we participants complete an imaged tactile pleasantness task. So we were asking participants to just think about being touched by either soft or rough materials on their own skin, and then to tell us how pleasant they think it would have been to be touched by these materials. So we wanted to have a measure of top-down beliefs about the pleasantness of the touch. In the second part, participants were completing a classic affective touch protocol. So they were blindfolded. We were delivering the touch with a brush at 5 different velocities, and we were asking participants how pleasant was the touch. So what we found is that both people with anorexia nervosa and people who have recovered from anorexia nervosa perceived the anticipated tactile experiences and rated the delivered tactile stimuli as less pleasant as compared to healthy controls. So we found a similar performance in the two clinical groups. However, this impairment had no relationship with the CT optimality. So in this second study, we didn't find this specific CT aspect of the effect. However, the differences in the perception of CT optimal touch were predicted by differences in top-down beliefs, so the first part of the task, in alexithymia as measured by means of the Toronto Alexithymia Scale, and interoceptive sensibility as measured by means of the Body Awareness Questionnaire. So with the second study, we concluded that the distortion in the perception of touch, or tactile anhedonia, if you want, there is an aspect that is more related as a trait rather than a state associated to the condition of malnutrition, and it tends to persist even after an otherwise successful recovery. Now, why is it important to target this aspect of anorexia nervosa? We think it's important because we know that people with anorexia nervosa, they show an impairment in multisensory integration. So we know that in order to create a coherent sense of our body, we do need to integrate signals coming from outside our body, so the so-called exteroceptive signals, with signals coming from inside our body, so the so-called interoceptive information. And what we see in people with anorexia nervosa is an imbalancement in this integration, in the sense that they tend to pay a lot of attention to exteroceptive signals, while they tend to downregulate interoceptive signals, as has been shown by other studies showing an impairment in the perception of pain, taste, and as we show also affective touch. What this means is also that we can observe a malleable body representation in people with anorexia nervosa, as has been supported by studies showing that they have a very strong visual capture in the rubber hand illusion. So they tend to acquire ownership of the rubber hand just by looking at it. And they also tend to have higher score of embodiment in the asynchronous condition, which is the control condition. They're not meant to experience any embodiment towards the rubber hand. This also means that virtual reality techniques could potentially be a promising avenue to alter body image in anorexia nervosa. And I'm aware that there are groups working on this, for example, the Chris Jigerman group in Utrecht, but also the group from Giuseppe Riva. But overall, these findings suggest that body image distortions in anorexia nervosa, they're not a state phenomenon that appear only during the acute state, so associated to a situation of malnutrition, but is rather a trait that persists even after otherwise successful recovery. Our findings about the perception of affective touch in anorexia nervosa, they fit quite well with a general idea that there is a strong link between affective touch and mental health. Other research groups have also put forward this idea, and here I'm just showing you some of the many studies that are out there linking investigating the perception of affective touch in relation to autistic traits, for example, or in relation to chronic pain. And our own group did a lot of work on the perception of affective touch in anosognosia for hemiplegia following right hemisphere stroke. And we found that there is a disruption in the perception of affective touch, and this is linked to specifically to damage to the right insula. Now, what can be done to address these issues of the perception of affective touch in a clinical population? Well, we can look also into the neurobiology of touch, for example, as we know that from the neurobiological point of view, positive tactile experiences such as hugging, massages, and seems to be related to the release of oxytocin. It's been hypothesized that oxytocin can provide the motivation to initiate and maintain tactile interactions, and it also promotes social interactions and well-being. We know that during interactions between mother and infants, or positive interaction between adults, or also between humans and animals, oxytocin is released in the body. But what is oxytocin? Oxytocin is a neuropeptide which is synthesized in the hypothalamus, and it has a dual function because it acts as a hormone peripherally in the body, and is related to processes such as labor and breastfeeding. But it also acts as a neuromodulator centrally in the brain, and is associated to the onset of maternal behavior. The development of a nasal spray allows the safe, non-invasive central administration of oxytocin to the human brain in order to study directly the behavioral effects. And we know that when we use intranasal oxytocin in doses between 18 and 40 international units, no side effects have been reported, which are specifically associated to oxytocin. One question that one could have is whether intranasal oxytocin is successful to reach the brain, if it can go through the blood-brain barrier, and the answer is yes. There are these important studies where Katerina was involved, and that show that intranasal oxytocin is successful in reaching the brain. And a particular like this study that they show they really analyze the active window of oxytocin through time, and we can see that the active window is until 35 to 40 minutes after intake. So this gives us quite a good timing to run behavioral experiments. Now, why we should associate oxytocin to anorexia? Because the oxytocinergic system has been proposed as a pathophysiological candidate for anorexia nervosa, and there is a cascade of studies that try to investigate the effect of intranasal oxytocin in various aspects of the disorders, such as food intake or the interpretation of facial expressions and emotions. And all these studies, they started around 2013 and are still running now. However, looking into the literature, there is really a lack of investigation on an important aspect of the disorder, which is this perception of interoceptive signals. So we really wanted to investigate whether intranasal oxytocin could be promising in improving the perception of interoceptive signals in anorexia nervosa. So this is a massive study. I'm just going to give you a little sneak peek of some of the results. So we ran this randomized, double-blind, placebo-controlled crossover study on 35 people with anorexia nervosa, restrictive type only, and 36 healthy controls. And the study was crossover, meaning that participants were receiving both oxytocin and placebo. So they were coming twice. 1 to 3 days, the two sessions were 1 to 3 days apart, and I was blind, they were blind, so we didn't know what they were receiving. And in the active window, the oxytocin, we completed the affective touch task, the interoceptive accuracy, and rubber hand illusion, but today I'm just going to talk briefly about the affective touch task. And if you are interested in this study, we published the full protocol a few years ago in BMJ Open. So the study procedure, everything is fully pre-registered. What we found is we replicate our previous findings. So main effector group, in the sense that people with anorexia nervosa perceive touch as less pleasant compared to healthy controls. And main effector velocity, so slow touch was always perceived as more pleasant as compared to fast touch. However, we didn't find any effect of oxytocin on pleasance or obsession, so meaning that there was no difference between pre and post nasal spray intake. Were we surprised? Not much, in the sense that previous studies, there is a bit of debate about the effect of oxytocin. For example, these studies from 2014 from Leckness groups, they found that intranasal oxytocin does not specifically modulate the subjective pleasantness of touch. However, this study just came out very recently showing that oxytocin increased the pleasantness of affective touch. So it's a bit difficult to understand what's going on with the effect of oxytocin. If we look in previous literature, we know that the effect of oxytocin, the effects of oxytocin are not straightforward. So, for example, this previous study showed that oxytocin selectively increased perceived pleasantness and the activity of the orbitofrontal cortex and insula only on male subjects when they believed that they were touched by a female experimenter, but not if they believed they were touched by a male experimenter. These other studies show that neural effects of oxytocin were found when subjects thought that they were touched by their partner, but not if they thought they were touched by a stranger. And again, the effects are super sex dependent. The study that just came out suggesting an effect of intranasal oxytocin on affective touch tested only male. We only tested female. So overall, these findings are in line with this idea that the effect of oxytocin on human behavior are highly context dependent. And we also know the beliefs about the nature of the compound matter. So if people believe that they received oxytocin or placebo, this can affect the result. So the next steps in this analysis of this huge database is to see if the beliefs of the compounds could affect the way in which oxytocin affect the perception of touch. And I will analyze this soon. To conclude, I've showed you that affective touch might play a really important role in the way we become aware of our body as our own. And people with anorexia nervosa present a disorder city-based affective touch system, and we have discussed implications for body image distortions in our first studies. However, in our second study, we showed that people with and recover from anorexia nervosa perceive touch in a similar fashion, and this tactile anhedonia seems to be a trait that persists even after recovery. And also, this distortion seems not to be related to the CT system, but rather a learned defective top-down anticipation of tactile pleasantness. So it's the learned aspect of touch that could be impaired. I also showed you some preliminary results from this first study to systematically investigate the effect of oxytocin in anorexia at different levels of social affective functioning and body-related awareness. And if you're interested, I also wrote an article for HAN, talk about the importance of touch for us as human beings and for our psychological and mental well-being. And with this, I conclude, and I just would like to thank my PhD and first postdoc supervisors, Paul Jenkins and Katerina Fotopoulou, and my current boss, Eric, all the patients who took part to our studies, and the funding bodies of our project, and you for your attention.
Thank you so much, Laura. Very amazing talk. So now we have time, 10 minutes for questions. There are no questions in the chat, so maybe I take the opportunity to ask you a question. Oh, okay, no. There is a question from Federica Scarpina. So thank you, Laura, for your work. I would like to ask if in anorexia nervosa, the baseline level of oxytocin may be reduced or increased.
Yeah, that's a very good question. There is the hypothesis that they might be reduced, and of course the oxytocin, the more we give oxytocin, the more oxytocin is produced, but the idea is that at baseline they might have a lower level of oxytocin. However, we cannot say if this was the case in our sample because we did not collect a baseline measure of oxytocin, which usually is done by taking blood or saliva samples, but we did not collect this data in this sample, so we cannot really say if this was the case with this in this study.
Okay, thank you. Thank you. I was thinking about the tactile anhedonia. So you found tactile anhedonia in patients with anorexia. So maybe do you think that tactile anhedonia in patients with anorexia could be explained according to a predictive coding frameworks? So maybe there could be something wrong at the beginning of the process or at the end, maybe on the reduction of the error. So maybe do you think that it could be explained according to this framework or not? So it's just a curiosity.
It's a challenging question. For what we can tell from our data is that definitely the priors beliefs about the touch matter. So the idea that the anticipation of how pleasant something will be, it really affects the way then they're going to perceive the touch. So in that sense, I think we could potentially use predictive coding framework to understand tactile anhedonia. But another thing that I think is also important is that we know that eating disorder, in particular anorexia nervosa, they have very high comorbidities with other psychiatric traits, for example, depression and anxiety. And it's often been suggested that this also when we submitted the paper, the review made a very good point that whether the effect could have been related to high level of depression and anxiety we observed. But we did run correlation analysis with the measure we had, and we did actually find correlation between these aspects. But definitely, I think the beliefs they have and also how they learned to perceive touch and the past experiences, probably they might play a role in the way they perceive touch on a daily basis.
Okay, thank you. So we have another question from Maria Alessandra Umiltà. So there are studies about the effect of oxytocin in another eating disorders. So could be its effect dose dependent?
Okay, so for the first question, another eating disorder, not that I can think of, or maybe I'm just being very narrow. I just looked at anorexia nervosa. It's also true that even in the study that they looked at only anorexia nervosa, is so heterogenic as a disorder that you have to consider many aspects. So, for example, in all our studies, we always look at only restrictive type anorexia nervosa, because already the restricted, the binge, the purging, they are different. They have a different symptomatology. So I think it's very important to just be precise in the recruitment of this kind of population. Effect dose dependent. So we actually used the highest dose possible, which was 40 international units. I mean, possible in the sense in the sense of past studies testing the safety. So I'm not sure whether it could be if we would give less oxytocin, if we would see a different effect. But I would think that it would be the other way around. That if we had used a very low dose, maybe we would not be able to see the effect. But I think with this dose, we should have been able to see an effect if it was there. But again, it could be that really if we take into consideration many individual differences and since the effect of oxytocin are so context dependent and there are so many individual differences, I think a more deep analysis might provide us with more information.
Okay, thank you. Okay, so just last questions from me. So I would like to ask you if you could explain more about one of your first study, maybe or second, that you presented today in your talk. So you said that you used visual stimuli, so face, if I correctly remember. So maybe if you want to explain more.
Yeah, so
I was interested.
Sure. So in this paper of 2016, while participants were perceiving touch, we were asked them to look at a computer screen showing either accepting or rejecting faces, so accepting smiling or rejecting faces or neutral faces. And we wanted to see if the facial expression would have influenced the perception of the touch. And we did this because we know from previous research that people with anorexia nervosa, they have an attentional bias towards rejecting faces, in the sense that they tend to ignore accepting smiley faces, and they tend to get stuck a little bit with rejecting faces. But however, we didn't find, we found that the effect of facial expression was the same in both patients and control. In the sense, in both cases, we saw that perceiving touch while looking at an accepting smiley face increased the perception of tactile pleasantness. So it was really nice to see this effect also in people with anorexia nervosa.
That's very nice, very interesting. So thank you. The last questions is from Susanna Walker. So great talk, Laura. Thank you. Do you have any feeling for why sometimes you see a velocity specific effect on the blunted pleasantness ratings in anorexia nervosa and sometimes don't?
Good question. I think generally what we see is that, also as has been more stressed from our second study in 2021, is that people with anorexia nervosa, they tend to perceive pleasantness in a more flat way. So they tend to differentiate less between CT and non-CT touch. What we do to conduct analysis is often we compute this affective touch index, which is the difference between perception between CT and non-CT, to see how good people are in differentiating between affective and non-affective touch. And what we see in people with anorexia nervosa is they tend to, this index is smaller, so they tend to perceive similarly affective touch and non-affective touch. As we said, as I said also before, the why is a bit difficult to understand, if it's something that they learn throughout life, or either or whether they're not being touched that much in their life and as well, so they don't value maybe this aspect of interaction that much. So it's difficult to tell, but our study with recovered patients probably suggests that it's something that is there before the development of the disorder, right? And is really a trait that is not associated with the malnutrition. So it could also be down to individual differences in perception. I don't know if it answered your question, but very good questions, Donna.
Yes, so thank you so much, Laura. So thank you again.
Thank you. Thanks.
Thank you. Bye. So